So first we'll talk about how DNA is replicated. This is what's going to happen during the cell cycle, during that interphase, during synthesis, when we go from one copy of DNA to two copies of DNA. So in order to understand DNA replication, you need to know complementary base pairing. This is the rule that adenine always pairs with thymine, and guanine always pairs with cytosine. This is the mechanism that our body uses to predict what nucleic acid it needs to put next in a DNA strand. So Watson and Crick actually predicted that this complementary base pairing would give the DNA kind of a mechanism by which it could be replicated easily. And then later on, another group of scientists called Michelson and Stahl did experiments and confirmed that this was indeed how DNA was replicated. So in the picture again, you can see that G bonds with C and T bonds with A. So imagine I only gave you the strand on the left. Would you be able to come up with a strand on the right? Yeah, because if you had a G here, you would know that this needs to be a C. And if you had a T here, you would know that this needs to be an A. And so that's the principle we're going to use, where we're going to use one strand as a template and then make another strand based off of it. So this is called the semiconservative model, where we take the parent strand, which in this image is colored in blue, and we're going to use it to create two daughter strands. And now these two daughter strands are going to have to be exactly identical to the original parent strand. And you can see how in the two daughter strands, half of it is made up of the original parent strand, and half of it's made up of these new nucleotides that we added, which are gray. And so this is semi-conservative, where each of the new daughter cells has half of the original and half new. So the first step in this is that we have to split the parent strand apart. When it's together like this, we can't add any new nucleotides. So we're going to take the molecule and split it into two separate strands. Then each of the two separate strands are going to get new nucleotides added using complementary base pairing rules. So I have an A here, I'll add a T. I have a C here, I'll add a G. I have a G here, I'll add a C. Notice that they used shapes to show how G and C can only fit with each other, and A and T can only fit with each other. After we've added new nucleotides to the entire length of the strand, then we have two identical daughter cells. So humans have 46 chromosomes, and our chromosomes are really, really big. So it doesn't make a lot of sense for us to start replication at one end of the chromosome and work all the way down. That would take a very long time. So instead, we have many sites along the chromosome. Imagine this one strand is a whole chromosome of DNA. So we have many sites along here called the origins of replication. And we start replication at all of these sites at the same time so that the DNA replication can proceed in both directions from each site, and the DNA can be replicated very quickly. So these create what's called a replication bubble, which is a small area of the DNA that has been separated. And the new DNA, that's the gray one, is being added in both directions at the same time. So as this proceeds, the bubble is going to get bigger and bigger until these bubbles eventually merge and we'll have two completely separate strands of DNA. The two DNA strands are in a shape called anti-parallel. When we talk about the ends of DNA, we talk about 5' and 3'. Notice that the 5' end is the end with the phosphate on it. We name it this just because it's on the number 5 carbon. It doesn't really mean anything. Notice over here, though, that the 5' end is also the one with the phosphate on it. Over here, we have the 3' end, and that's the one with this hydroxyl group, no phosphate. And then over here, we have the 3' end again. That's got the hydroxyl group with no phosphate. So these two DNA strands run in opposite directions. This one runs 5' to 3' going down, and this one runs 5' to 3' going up. So this is what gives our DNA this anti-parallel structure. So here are some enzymes that are involved in DNA replication. Once the parent strand is split open, right, the very first thing we have to do is open up the double strand. Then we have a molecule called DNA polymerase. And this enzyme can add nucleotides to the new growing DNA strand. One thing that's interesting about this is that it can only go in the 5' to 3' direction. So if you look at this replication fork, which is part of a bigger replication bubble, this strand over here is being synthesized continuously. It's going from 5' to 3' towards the replication fork. The other strand, remember, DNA is anti-parallel, so in order for DNA polymerase to go 5' to 3', it has to make little short segments going in the opposite direction, and then when the fork opens up again, it'll be able to make another little segment. So because DNA is built in segments, we need to have a way to put the two DNA segments back together. So here you can see two of the segments are broken apart still, and so we have another enzyme called DNA Ligase, which comes in and takes these two segments and connects them back together. Once we have a new piece of DNA, then we have to proofread it. Think about typing in Microsoft Word. You still have to go back and edit your paper before you're done. So once we've copied the DNA, DNA polymerase goes back and rereads through all of the pairs to make sure that A is paired with T and G is paired with C.